Hey guys, welcome to Chapter 8. In Chapter 8 we have 3 different topics, and each of these are going to be pretty small topics, so hopefully they'll be really good manageable bites. This chapter is directly related to Chapter 7 where we talked about how we make gene products. Now we're going to talk about how we regulate gene expression, because obviously we can't let the cells just go making whatever proteins they want willy-nilly. So we're going to talk about all of those things and how they work, how transcriptional switches work, and the mechanisms that develop specialized cells. This first topic is just a real quick overview, so hopefully this is nothing new in this first topic, but make sure you pay attention because it is important material. So in this topic, we're going to talk about how organisms develop into different cell types. We'll talk about unique cell products. We're going to talk about the impact of external signals and the checkpoints in gene expression. As always, here's our topic objectives. Please make sure you go through these and let me know if you have any questions about them. There's a lot of objectives for this very small topic we have here for the first part of Chapter 8. So make sure you go through them. And as I said, let me know if you have any questions. So first of all, gene expression is very important for the health of our cells and us as an organism. It's really important that we have gene expression. Otherwise, when gene expression gets out of control, we lose, or this is when cancer develops and other deadly diseases develop. So gene expression is really, really critical. So let's take a moment and let's define 2 quick terms. And these are really important for moving forward. The first is gene expression. And gene expression is the process where a gene affects an organism by directing the synthesis of a protein or an RNA molecule with a specific activity. What does this mean in layman's terms? It just basically means that the process of this gene is somehow being controlled by something else. That's all it means. It means that it's either preventing the synthesis of a protein or an RNA molecule, or it's doing some kind of specific activity to control that. And then cell differentiation is another term we need to understand, especially because we're going to talk about here in the next slide, is that cell differentiation is the process where a cell goes through a change to become a specialized and easily recognizable cell. All the cells in our bodies are very different from each other. If you were to take a sample of a skin cell, look at it under a microscope compared to a brain cell or a nerve cell, it's going to look different. And this is because of cell differentiation. So let's look at that now. So as I said, there's lots of different types of cells in the human body. And at fertilization, we all start out the same, right? Egg and sperm meet to form the zygote, and all those cells from then on for a while look identical. And that's because they haven't been differentiated. This differentiation begins during embryogenesis, and there's a whole series of genes that actually control this. And so it's important that you understand that. I don't care if you know which specific genes, but just be clear that you understand that that's how that works. And that's actually why we can't just regrow organs when we need them, is because we aren't quite sure how to get these cell types that we need in the specific way. And this is all done through the regulation of proteins. And before this differentiation, these cells are all known as embryonic stem cells. And this is where we get into that argument about stem cells and how they can be used. And I know that you have heard of adult stem cells as well. And these are cells that are, like, for instance, in our bone marrow that are undifferentiated as well. And that's where the term stem cell is arising from, is it's not a differentiated cell. So these cells, though, even though they're undifferentiated, and even the differentiated ones will always carry specific information within them. And you can see how this works when we talk about cloning. We talked a little bit about cloning earlier in this unit. But what happens is if we were to take a differentiated cell from any organism and put it into a unfertilized ovum and allow that cell to develop into a new organism, we're going to grow the exact same organism that we took the DNA from the first took the information from the first differentiated cell. So that's really important to understand, that is that the material is already there to say what it is, and it's going to use that over and over and over again to create the same organism. You can't necessarily take frog DNA and a frog nucleus and make it into a cow. For instance, on this slide, as you can see, we start with one organism, we're going to finish with that same organism. And we're not going to go too much into cloning, at least right now especially, but the big key to here is understand is that by taking out the genetic information and putting it into something that's unfertilized and carrying that forward, you're going to see the same set of instructions carried forward to create differentiation. Now, this just kind of shows us that they are different within the same organism. Not only do we have this different instructions between you and your dog, but you also have different instructions between your brain cells and your liver cells. And we've talked about this a lot. We've talked about this assay, especially with regards to the different cell types. And we've talked about the 2D Western blots, and that's exactly what you can see here. You can take the different proteins from the human brain and the human liver, and by doing the two-dimensional focusing, or the two-dimensional Western blot, which remember, you do one dimension is pH, one dimension is size, which gives us these dots rather than bands. And you can see here how the proteins of the human brain are very different than the proteins of the human liver. And this is all through differentiation and gene regulation. And this is all through the housekeeping genes and all these things that keep these cells different from each other. And that's really what I'm trying to get at here for this entire topic for you, is to realize that even though these cells are all different, they look different, they act different, there's different gene regulation. And this is why gene regulation is such a big deal, because if we can understand how this works, we can either regrow organs, we can cure diseases. There's a whole variety of things that can open the minute we understand gene regulation. And we're only just touching on the surface of that right now. And the last thing to talk about within this topic is that even though internal regulation is the main driver, external regulation plays a big role as well. Based on what's going on outside the cell, these cells are all communicating with each other. And based on outside signals, these outside signals are going to tell the cell to do something it wouldn't normally do or react differently. And so it can increase gene expression, it can decrease gene expression. So external factors do play a bigger role in this as well. Maybe not as much as internal, we could debate that in class, but the big thing to understand is that it's not just a vacuum that these cells are working in, and that there are outside things that can change the way they work. And this is an example you can see on the slide here of a liver cell, and with the glucocorticoid hormone being produced. And you can see how it's beginning the production of the enzymes to specifically break down proteins into glucose for the cell's body. And that's when we don't have enough glucose in the area. So it's important to understand how that can work. And I don't want you to memorize this flowchart here or anything, but just be aware that that's how these protein and gene expressions can work. It can be as small as changing the production of hormones from the liver. It can be as big as differentiating cells in an embryo. So this is the end of this topic. Hopefully most of this was just good review or good information. And when you're ready, go on to topic number 2.